All right, team. Today we move into a new unit and we begin with studying bacteria and viruses. And then we also do, I believe, the kingdom Protista in this unit. So this is, I mean, for me, this is kind of the fun part of the class. I wish we could all be together, but I think you're going to really enjoy learning about these organisms. We're doing kingdom surveys. It will include life cycles once we get into the eukaryotes. Prokaryotes, the bacteria, don't really have very complicated life cycles at all. They don't sexually reproduce. And the viruses, of course, aren't even living organisms. So where we are is in module 3, and you can see module 3 says bacteria, and if you click on the bacteria and virus PowerPoint, that's where we will be today. Okay. And so we break this up into domains. Now, there's a lot of confusion, or there can be confusion about the taxonomy here. So you know, just to be clear, we have 3 domains now, and a domain is actually a level above kingdom. We no longer classify the bacteria in kingdoms. We used to include all bacteria, all prokaryotes in kingdom Monera. So we want to spend a second talking about what it means to be a prokaryote as well, because I don't think we've done that. The third, just so you know, the third domain is the eukaryota. And of course, that's not what we're studying now, but this includes everything else. So we put the bacteria in 2 domains, the archaea and the bacteria. Archaea are very primitive organisms, the first known life forms on Earth, most likely. Kind of an oddball group of organisms, which we'll talk about. And then the vast majority of bacteria, you know, in quotes, are in the domain bacteria. So when I use the word bacteria, I'm using it generally to refer to all the prokaryotic organisms, including both of these domains. And I know that can be a little bit confusing. Okay. So what does it mean to be a prokaryote? No nucleus, right? No membrane-bound organelles. And generally speaking, prokaryotes are smaller, as small as 0.2 microns. Is micron. So that's very small, right? They can go all the way up to, you know, much larger than that. I'd say on average, they're 2 to 4 microns on average, okay? So that gives you an idea of what we're going to be studying. Again, the archaea are kind of oddball group. We'll talk about 3 different sort of subgroups of the domain archaea, and then the rest of our discussion will be talking about the bacteria, the domain bacteria, which includes all of the organisms which are known pathogens, right? Okay. So now we've already seen this taxonomy. What is interesting facts about the bacteria? Well, although they're small, although they're tiny and we can't see them with our naked eyes, it turns out that the bacteria as a group of organisms is kind of running the show, y'all, believe it or not. They are kind of running the show. Just to give you an idea of their just incredible abundance. If you took a gram of dirt, so think about like an equal packet, that's a gram of powder or a sugar packet. If you just went to an agricultural field, let's say a farm, and you took a gram of dirt and you counted the bacteria, there would be 40 billion with a B bacteria in 1 gram of dirt. It's a tremendous amount of bacteria. So that kind of gives you an idea. So you think about a pound of dirt, think about a whole field, how many, just unbelievable numbers of bacteria. So if that's not impressive to you, if you could somehow round all the bacteria that lived on the earth, if you could round them all up and you could somehow put them on a scale, right? And on the other side of the scale, you put all other life, all the plants, all the animals, all the fungi, all the protists, all the other living things on the other side of the scale. The bacteria would weigh more, have greater biomass than all other living things combined. So, this gives you an idea of how incredibly important they must be to the ecology of the earth, right? We have in our bodies about 60 trillion cells. And that's, obviously an estimate. So if you look at a human being and you counted up all of the human being cells, there would be about 60 trillion cells, give or take a trillion, right? Living in and on those 60 trillion cells are 600 trillion bacteria. That's right, folks. There are 600 trillion bacteria living in and on your body, all over your skin, all in your gastrointestinal tract. And we already talked about how you can't really live without the bacteria. They're an important part of the symbiotic relationship that we have. Okay. So those are just some interesting, startling facts about bacteria. I forgot to set my timer. Let's do that. Okay, so what else can I tell you about them? There are many different ways to classify the bacteria. And now at this point, we're really talking more about the domain bacteria. Again, we're going to get to the archaea, the very primitive ones, in a bit. One of the ways that you can classify them is based on their shape. It's pretty straightforward. We have 3 different shapes that we recognize in bacteria. Now, at this point, we need to say a couple of words about heterotrophic versus autotrophic. We really need to understand what these words mean in order to continue our discussion, and then we'll talk about the shapes. So heterotrophic, it literally means you can't make sugar. Right? Autotrophic means you can make sugar. How do autotrophs make sugar? Well, the vast majority of them do it through photosynthesis, right? But there's also this oddball thing called chemosynthesis. We didn't talk much about it. We haven't talked at all about it, really. And, now's the time, because we're going to see that some of the archaea are actually chemosynthetic. Chemosynthetic organisms use heat instead of light as the energy source, and they use H2S, which is hydrogen sulfide, instead of water as the electron donor. And these organisms, we'll talk more about them. They live typically in places that are very hot, like hot pools or heat vents at the bottom of the ocean. We'll talk more about these in a bit. But if you're autotrophic, those are really the two ways that you can make sugar, through photosynthesis or chemosynthesis. Heterotrophic organisms cannot make sugar. Okay. What about us? Well, we're heterotrophic. So what does it mean if you're heterotrophic? Well, you have some choices in terms of how you can make your living. Let me make a little more room here. We just want to focus on heterotrophic because we talked about autotrophic. We want to focus on heterotrophic. What we know is there are different kinds of heterotrophs. For example, you can be saprobic, and saprobic organisms are a type of heterotrophic organism that eats dead stuff. These are the decomposers, right? Of which many bacteria are, along with the fungi, right? But you could also be parasitic. And if you're parasitic, you make your living by eating live stuff. And there's safe to say there are lots of bacteria that are parasitic, including ones that are parasitic on us, right? So what I want to tell you is that it turns out when you're talking about bacteria, there are both autotrophic and heterotrophic bacteria. And when we're doing this classification thing using shapes, these only refer to the heterotrophic bacteria. We're going to get to the autotrophic bacteria later, but we won't be able to apply these terms to them because they're a little bit different. Okay, so I want to make sure everybody's clear on all that before we move on. Okay. All right. So we have cocci, which are spherical or round. You can think of them as round, and we'll look at some pictures of these. Bacilli are rod-shaped bacteria. And you have to watch out because sometimes these things they'll kind of stick together like this. But here's the thing, all heterotrophic bacteria are unicellular. This is very important to understand. Each cell is an individual bacteria in the case of the heterotrophic bacteria. Now, what we're going to see in the autotrophic bacteria is sort of an advancement in life form. And when I say they're unicellular, this is an example of a life form. You know, if I ask you what's the life form of a heterotrophic bacteria, you would say, well, they're all unicellular. Some other life forms might be colonial or filamentous, and we'll see examples of those later. Okay. So sometimes these rods will kind of stick together, and you know that might give you the impression like they're actually multicellular, but they're not. They're just stuck together. Okay. And then we have sporili, which are like corkscrews, like this. The sporili are some of the most advanced of all the bacteria, and we see. You know, a lot of the sporili are flagellate, so they can actually swim rather than just wiggle. Okay. So let's look at some pictures of these. And then we'll talk a little bit about the basic morphology of a bacteria. I should have probably already done that, but it doesn't really matter too much. Okay, so there's some cocci. You can see all these little balls. Each one of these little circles is an individual cocci bacteria. Okay. There are bacilli. I'm not convinced these aren't just Cheeto Puffs. They look like Cheeto Puffs to me. I don't know. I'm just saying. No, those are rod-shaped bacteria. You can see they're kind of funny looking in this picture. And then there are some sporulli. You can see they're kind of corkscrew. They're really more like three-dimensionally corkscrewed, believe it or not. Okay. Let's take a second to draw a bacteria. Whoa, look at this. Man, we're making proteins. That's from my general biology. Okay. Take a second to draw a bacteria. I was going to draw one. See, what's it made out of. We'll draw a rod-shaped one. And it does have a cell wall. It also has this interesting layer. It's called a gelatinous sheath. It's like this layer of slime. Sometimes it's called the slime layer. It's what makes bacteria sticky. And this is the gelatinous sheath. Or slime layer. Best example of that I can give you, maybe you stayed out a little too late one night, you got home, you were tired, you passed out in your bed without brushing your teeth. And then you wake up the next morning and you got that film on your teeth that's really slippery. Well, what you're feeling is the collective slime layer of all the bacteria that have colonized your teeth during the night. Okay. Now, remember, as prokaryotes, these organisms, as far as what the slime layer does, it allows it to adhere or stick to surfaces or to its friends, its other friendly bacteria. Okay. Now, as prokaryotes, there's no nucleus, but they do have to have genetic material, and this is a circular DNA. And it just is literally a piece of DNA molecule that attaches to its end. It attaches end to end. It makes a little circle. Okay. The only other thing you might see on these bacteria are these little finger-like projections, which are called pili. Pilus is singular. There's one pilus, or pili is plural. They can have those. And these are like little fingers of attachment. These allow them to attach to things. Or the more advanced ones can even have flagella, and they can swim. Okay. So that is about it. There's not a lot of internal structure aside from the DNA, and then we have the pili and the flagella. Not all bacteria have flagella, by the way, only the more advanced ones. Okay, so now you know a little more about the morphology as we continue. Another way to classify bacteria is by using a gram stain, and what this means, you've probably heard gram positive and gram negative bacteria. Right? And what these terms refer to is some bacteria have a chemical called peptoglycan. You see that word right here in their cell walls. And they developed a stain which would stain this peptoglycan, which is a carbohydrate, and it's called a gram stain. And so what you do is you take you make a culture of a bacteria and you apply this stain, and if the bacterial cell walls absorb the stain, then you would say that would be considered a positive, and you would say that was a gram positive bacteria. Or if it didn't absorb the stain, you would say it was a gram negative bacteria. Why is this helpful? Well, most Disease pathogens that are bacteria have peptoglycan and they're gram positive, for example, strep. So believe it or not, back when I was a kid, if I had a sore throat, my mother took me to the doctor. You know, they would swab your tonsils just like they do now, and then they would say, well, you know, we'll let you know in about a week whether or not you have a bacterial infection. And so literally what they would have to do was culture that bacteria, grow it out on a Petri plate, right? And then stain it with a gram stain once they grew it out to see if it took up the gram stain. And then that's how they would make the decision. Now, how long does it take? You go to the doctor with a sore throat, it takes a matter of minutes, right? They have much more sophisticated ways of testing than they had when I was a kid. Okay. At any rate, I'm sure there are countries in the world where they don't have that sophisticated method, and they're probably still using a gram stain to, you know, test for the presence of pathogenic bacteria. There are some gram-negative bacteria that are pathogens as well, so I don't want you to think that it's exclusive. Okay. Now we have a list. We have the sort of the short list of bacterial pathogens, right? Now we're in the domain bacteria, we're not in the domain archaea at this point. Everything really that we've talked about up to this point really refers to the domain bacteria. Okay. Well, bacterial pneumonia is one of the most common causes of death in older folks, and it is extremely common. Bacteria that normally live in your lungs can infect your lungs if you have an compromised immune system, or if you just, you know, if you're very old, your immune system is typically a little bit compromised. There are, you know, treatments obviously for most bacterial infections, right? What was the big game changer for bacterial infections? Antibiotics, right? And we'll talk a little more about antibiotics when we do the fungi, because it turns out that most of the antibiotics, the first antibiotic and many others, were derived from fungi. So suffice it to say that prior to the advent of antibiotics, particularly penicillin, if you got a bacterial infection like bacterial pneumonia, or maybe you got syphilis, which is a sexually transmitted bacteria, or you got tetanus or gangrene or, you know, salmonella or shigella poisoning, there was a pretty good chance you were going to die. Particularly skin infections were dangerous, like staph. You know, you could get a superficial flesh wound and it gets infected, and there's literally no way to treat it prior to antibiotics. And so many of these things, like syphilis and gonorrhea, for example, which are STDs, you know, you don't hear about them nearly as much as you used to back in the day before antibiotics. So turns out that we have now a super resistant strain, we'll put super strain, of syphilis, which is believed to have been brought back from Thailand as an unfortunate consequence of the Thai sex vacation trade, where people literally go to Thailand with the goal of having sex, sometimes with juveniles, or basically sex with prostitutes. And so apparently this group of men went there on a sex vacation, and they picked up this strain of syphilis, brought it back and passed it on to their wives and you know, significant others in this country, and then it spread from there. And this is a super mutant strain of syphilis for which no known antibiotic will cure. So that's a big deal, right? Very serious. And you know, we see antibiotic resistant strains of staph, of many other bacteria. So this is a consequence of misusing antibiotics primarily, and we'll talk more about that later. What about anthrax? Anthrax is one that you may have heard of. Anthrax and Legionnaire's disease actually are closely related in terms of their symptoms. Anthrax is fairly common as a skin disease, much like staph, and so it wouldn't be uncommon for example, a farmer who got scratched by some poultry like a chicken or a turkey to get a little skin lesion of anthrax. And, they would treat it with some antibiotics and it would generally resolve itself. However, if you breathe in the spores of anthrax in your lungs in a sufficient enough quantity, it can actually become systemic. It can invade through the lungs, get in your blood, and then it is about 80% death rate. So it's very serious disease. Now, many of you are too young to remember this perhaps, but shortly after 9/11, some wingnut was mailing anthrax in the mail. They figured out how to micronize the spores to where just opening an envelope would actually kick enough of these anthrax spores in the air to infect the person that was opening the mail. And several people died. He was mailing them to politicians and reporters and various other people. I say he, it sounds kind of like a he thing to do. I don't know. I suppose it could have been a she. And then all of a sudden it just stopped. And, I suspect that this individual maybe accidentally infected themselves, or maybe they just got bored. I don't know. They never did solve the case. No one knows who did this, but it was a scary time. Legionnaire's disease and anthrax, both, what happens is they become hemorrhagic. And hemorrhagic means you bleed out. It starts affecting blood vessels. Bubonic plague is the same way. So they called bubonic plague the Black Death, right? These are all hemorrhagic fevers, anthrax, Legionnaire's, and bubonic plague, meaning they weaken the blood capillaries and the blood just starts to leak out. And this, you could imagine, they called it the Black Death or the Black Plague because your whole body would turn black just from what is really a giant bruise underneath your skin from the blood leaking and coagulating. So it's not what you want your doctor to tell you. You got a case of hemorrhagic fever. Okay. Legionnaire's disease, a very curious one, very recently discovered. It was in the 80s that it was discovered. And they called it Legionnaire's disease because it was discovered among attendees to the American Legion Convention. The Foreign Legion these are veterans of foreign wars, and they have Legionnaire's conventions. They would have them once a year, and usually they would rent out a big hotel and a conference center. Well, in Philadelphia, they had it one year in the 80s, and people started getting really sick, and they didn't know what the heck was making them sick. They eventually, when they started dying, at first they thought they must have had food poisoning, but then they started dying, and they were like, whoa, what is this? They discovered a bacteria which had never been described before, which they named Legionella after the Legionnaires. And it is, similar to anthrax. It becomes systemic. It causes hemorrhagic fever. Since that time, it seems to disappear and then crop back up. It's one of these strange bacterial pathogens that we don't really know where it hides. Perhaps it hides in some other animal, and occasionally it will crop up in a nursing home or a hospital or someplace where people congregate, particularly people with compromised immune systems. Okay. What about tetanus? How do we get tetanus? Hopefully we don't get tetanus. You always hear, how do you get tetanus? You get it from rusty metal, right? Well, it doesn't have to be rusty metal, but any sort of a puncture wound that goes down, penetrates into the subcutaneous layer, into the underlying connective tissue, could potentially give you tetanus. And so people commonly get it in like a farm setting, if you were caught on some barbed wire or cut your foot on a nail or something like that. because there's a tetanus shot, which is good for I think they now have a 12-year shot. Tetanus is extremely uncommon in this country. There were only 66 cases last year of tetanus in the USA. So that makes it really rare. And so, tetany refers to the muscles becoming permanently contracted. This is a neurological disorder. Primarily, it attacks the neuromuscular junctions. The first nerve that it attacks is the vagus nerve, which is the one that innervates your jaw. And so it causes what they call lockjaw, where your mouth is closed and you can't open it. It could very quickly spread to the brain and kill you. So it's no joke, nothing to mess around with. If you're going to leave the country, get a tetanus shot before you leave. Why is this? Well, you don't want somebody in another country sticking a needle in you, most likely. You're better off relying on the folks here in the USA that where we have, we use sanitary practices, et cetera. Particularly if you're going to a third world country. Okay. Botulism and gas gangrene are related. They are both organisms in the same genus, and the genus is Clostridium. So botulism is caused by a bacteria called Clostridium botulinum. And what makes this genus Clostridium very interesting is that it is a group of organisms, a group of bacteria that are obligate anaerobes. What in the world does that mean? Well, if you're an anaerobe, you can live where there's no oxygen. And you're basically doing anaerobic respiration to make your energy, your ATP. If you are an obligate anaerobe, you can only live where there's no oxygen. If you put oxygen in the picture, the Clostridium botulinum organism can't grow. And so literally you could take a sterile swab and swab your house and then culture it in an anaerobic environment, and most likely there would be Clostridium spores and you could get them to grow in an anaerobic environment. But as long as there's oxygen, they can't grow. So botulism is actually a form of food poisoning. And I'm just going to tell you that modern canning has eliminated it from commercially canned food. You're probably, the chances of anyone getting botulism from a commercially canned can of food is almost zero, okay? Why do people still get botulism? Home canning. And the biggest culprits are low acid foods that are improperly canned. Example, beans and squash. And here's the situation. Depending upon what it is you're canning, you have to treat it differently. Just to give you an example, tomatoes, which are a very high acid food, you can pack those things in canning jars. You can do a 10 or 15 minute hot water bath. You don't even need a pressure cooker, just a hot water bath, and those will be safely preserved because they have a lot of acid in them, right? Compare that to canning string beans, where it takes 50 minutes at 15 pounds of pressure in a pressure canner to safely render that food edible, okay? The reason is that the botulinum spores are incredibly heat resistant, and they're not tolerant to acidic conditions, but they're very heat resistant. And so if you don't cook them enough, if you don't cook the beans enough, because you create this anaerobic environment by basically boiling the oxygen out of the water in the jar, you create this anaerobic environment. And if some of those spores have survived the heat treatment, they will begin to grow. Now, one of the things I'll tell you, a good practice when you're canning is once the cans have sealed, remove the bands, not the lids obviously, but the bands from the lids. This is a smart practice. You don't really need the bands because the lid is sealed on there. And what this will do is, if something does start to grow in your jar, it's going to be respiring. and it's going to be producing carbon dioxide, which is a gas, and eventually that CO2 will pop that lid off. And so if your lids of your canned goods all start popping off, you know something is growing in there. And so that's a really good practice. The most recent case of botulism I heard about was in Henderson County on Easter, and this has been about 5 years ago. Easter Sunday, a woman made 3-bean salad with some beans that she had canned. She poisoned her entire family. She almost died because she ate quite a few of them before she even made the salad. So here's the thing. The botulinum toxin, which you know of as Botox, right? Is incredibly toxic. They did this, they did these calculations with the pure botulinum toxin, and they said if you had about 5 kilograms, that's about 12 pounds of the pure botulinum toxin, and you could somehow divvy it up among every man, woman, and child on earth, you could kill them all. That's how toxic it is. So we measured toxic lethal doses of Botox in micrograms, very, very tiny amounts to kill someone. Yet we also inject Botox into people, both in the cosmetic industry it basically paralyzes the muscles, and so it gets rid of wrinkles essentially, because it paralyzes the muscles and tightens the skin that way. But anecdotally, they discovered it also seems to be very effective at treating migraines, and so now they use it to treat migraines, and they also use it to treat people that have incontinence. They can't control their bladder, and so they actually inject tiny, tiny amounts of the Botox into their bladders, and it helps them with incontinence. Okay. So if you're canning, use a modern day canner and a modern day canning book. The woman that poisoned her family, they said, well, tell us about what you did. And she said, well, I used a canning recipe from the 1940s, right? So that would have been before they really knew a lot about this, okay? Gangrene, now it's important to mention, Clostridium botulinum is not a parasite of humans, right? It's just that its waste products are extremely toxic, and so that's important to understand. It's not in and of itself a parasite, okay? Unlike the Clostridium species which causes gas gangrene, this is a flesh-eating bacteria, and it survives in the space between your connective tissue and your skin, which just happens to be a nice anaerobic environment. So if you got a puncture wound, a deep puncture wound, you could potentially get gangrene from it, or if you have frostbite, because you could imagine when the blood stops flowing into an extremity like your big toe, let's say, or your fingers, there's no oxygen. And so now we have an anaerobic environment and gangrene can begin to grow. Okay. Back in the Civil War, many, many, many soldiers died from gangrene because there was no treatment, there were no antibiotics. And so you might get just a superficial wound. Maybe you were just grazed on your arm by a Manet ball, by a lead ball, and it gets infected. You're fighting in a cow field. There's cow poop everywhere, right? It's easy for it to get infected. And then, so many, many, many more soldiers died in the Civil War from secondary infections than died from bayonets or bullets. Okay. What about the bubonic plague? Is that relevant? Well, let's think about the fact that it killed half of Europe dead, right? Roughly half of Europe died in the 11th, 12th, and 13th centuries from the bubonic plague. It does still exist on Earth, and occasionally it will crop up. But for the most part, it's under control. Now, I mentioned, or I may have mentioned, that there is a mutation that some humans have that makes them immune to the bubonic plague. And they always wondered about this back in the day because there were people, particularly priests and doctors, who were trying to help the sick. And they were exposed over and over and over, but they never got sick. And they thought it was just by the grace of God. But as it turns out, they had this mutation. This same mutation now protects their ancestors from HIV. And I believe we mentioned this when we talked about mutations in a previous lecture. Okay. So the Black Death, it is fairly complicated. It is a bacteria which lives in the gut of the rat flea. So if you're bitten by the rat flea, or you're bitten by the rat, or you're exposed to blood from anything that's been infected, you could potentially become infected. Okay. Tuberculosis is a bacterial infection of the lungs, which was much more common than it is now. It's because of screening, we've almost eliminated it in this country. Salmonella and Shigella are both foodborne or waterborne pathogens, which cause serious GI tract infections. Salmonella in particular can kill you. We have people that die every year in this country from salmonella poisoning, almost always from food that has been contaminated with salmonella. This bacteria is very common in chickens and poultry, and this is one of the reasons why you're supposed to make sure you cook poultry thoroughly. But interestingly, it's almost always the consumption of vegetable matter like spinach or strawberries that infect people with salmonella. And so you may be wondering, how in the world are these plants getting infected with this bacteria that lives in the gut of a chicken? Well, you might be surprised to know that the way it happens is through cross-contamination. And so it can be as simple as the dodo that loaded the produce truck or the food delivery truck put, the spinach on the bottom of the shelf of the truck and the chicken on the top, right? And then the chicken juice dripped out of the box onto the spinach, right? Cross-contamination. And then the chef was in a big hurry and didn't have time to wash the spinach thoroughly, and it ended up on the spinach salad plate. And then boom, you have cross-contamination. Same thing can happen in your home. Your roommate can cut up a chicken on the cutting board and forget to wash it. And then you cut up your romaine lettuce and you cross-contaminate it. So you got to really watch out for these types of things. Okay. Typhoid fever, very serious illness, very common during times of natural disasters or wars, or just in third world countries where there's just not a lot of sanitation. Okay. Leprosy, wow, we've almost eliminated it. It's not super contagious. Crazy thing, y'all, the armadillo has made it to North Carolina. I don't know if you're aware of this. And armadillos are capable of carrying leprosy. So they tell you, do not mess with armadillos, just leave them alone, because you could potentially get leprosy. Now, with modern antibiotics, we're able to treat this disease. Okay, but even so, you don't want to go there. Okay, so that's a pretty darn good that's the short list of bacterial pathogens, and you could imagine we're leaving a bunch out, right? But that's a pretty good list. Okay, hopefully you found that interesting. What about the relevance of the bacteria to global ecology? And what does that even mean exactly? Well, what we're saying is, what do these things do globally? Well, globally, they are the decomposers of the earth along with the fungi. They also produce tremendous amounts of carbon dioxide. Remember, they're the most abundant life forms on earth, right? And it turns out that because the vast majority of bacteria are heterotrophic, that along with the fungi, they produce an astounding amount of carbon dioxide, up to 80% in a given year of the carbon dioxide that's liberated on Earth is liberated by bacteria and fungi decomposing things. And so there are people that say, well, see, we shouldn't even hold ourselves accountable over this global warming CO2 thing because it's not even us, it's those darn bacteria and fungi. Well, our land use practices have a direct effect on this number, right? When you cut all the trees down and pile them up in a pile and let them rot, you're creating food for bacteria and fungi, right? So certainly we have an impact on this number here. And then as decomposers, they help to recycle dead things. Like think about the leaf litter, all those billions of leaves that are falling off the trees get decomposed and returned back to their constituent components back into the soil. So that's a good thing. Okay. Maybe we'll do one more slide. Actually, yeah, we can make it through this. So we can talk about the domain Archaea now. These are oddball groups of organisms. I'm just going to tell you that. They have very specific niches in nature. And you could imagine what they are. Thermophilic must love what? Heat, right? These are heat lovers. These bacteria live in hot pools. They live in natural hot pools or hot springs. They live in heat vents at the bottom of the ocean. And many of the thermophilic bacteria are chemosynthetic, which is interesting. They're the oddball things. Halos in Latin means salt, and these are actually salt lovers, which is odd, right? They live in very, very saline environments. And I mean many times more saline than the ocean. And then the methanogens are methane producing bacteria, which live in mostly anaerobic environments, and they're able to produce methane as a byproduct, methane gas. And so we'll look at some pictures and talk about each of these just briefly, and that will literally be the only thing we'll have to say about the thermophilic bacteria. Okay. I'm sorry, about the domain Archaea. So what you're looking at here for perspective is Yellowstone. If you've never been to Yellowstone, before you die, and I hope you don't die soon, but before you die, go check out Yellowstone, or before it explodes, right? It's very unstable geothermally speaking, right? What you're looking at, this is the paint pool. This is a very famous place. And what you're seeing are thermophilic bacteria. This water is very, very hot. It's steaming hot. And from here to the middle, these different colors that you're seeing are different concentrations of different kinds of thermophilic bacteria. The rusty ones live on the edge. It's actually hotter in the middle than it is on the edges. And so that's why we see these different colors. You can see the yellow in between. Just for perspective, that's a person right there on a walkway. This walkway is probably 6 feet wide. And so you get an idea of how big this pool is. This is just one of many of the fabulous things in Yellowstone. Now, again, these organisms typically are chemosynthetic. They use heat and hydrogen sulfide as a they use heat as an energy source and hydrogen sulfide as an electron donor instead of water, and they give off sulfur. And so these pools typically have kind of a sulfury, like a rotten egg smell, and that's what you're smelling, is the sulfur that's being released as a byproduct of chemosynthesis. These are halophytic bacteria. For perspective, what you're seeing here is a canal. This is in Portugal. This is salt right here on the edges. It looks like sand or snow, but it's actually salt. And what they do is they flood these canals with seawater from the Mediterranean, and then they let the water evaporate. And as the water evaporates, we have the salt crystals forming, and they harvest it and sell it. But if you notice in here where the water is, has this pink color. And this is due to the presence of these halophytic bacteria. Now, the bacteria are harmless, and it used to be they would bleach the salt. Sometimes they would use chlorine to bleach it, and you could even taste the chlorine in the salt. Now, the different colored salts are all the rage, right? Like the Himalayan pink, and if you buy Celtic sea salt, it can be gray or brown. This is just due to the presence of these halophytic bacteria. They're harmless to us. But they do discolor the salt. And so if you think your salt's supposed to be white, you might want to avoid these products. Otherwise, they're fine. Okay. They're expensive. For some reason, leaving the step of bleaching out makes it more expensive. I don't get it, but that's how it is. Okay. Last slide we'll do today is a slide that deals with the methanogens. And this isn't really all. I don't really think that he's passing gas and lighting it on fire, but it's cute, right? So we do actually, believe it or not, have methane producing bacteria in us, in our gut, which is fairly anaerobic. These organisms can live, and we can pass some stinky gas sometimes. And you're smelling the methane. Methane has, again, kind of a rotten, eggy smell to it. Okay. Where we find these things is in anaerobic environments. Old landfills are a really good place to find them. You know, you have a landfill, you dump all your garbage in the ground for years and years and years, and then at some point they're like, okay, it's full. And then they put a cap of earth over it, right? And then, you know, they'll build a little playground or something. It used to be, there was the perpetual torch. If you pass by an old landfill, you could see there was a flame, there were these pipes sticking up and there would be a flame. And what they're doing is they're burning off the methane gas that accumulates. Now, when they go to design the landfill, they actually dig wells, like gas wells, into the landfill as they're filling it, and then they actually siphon off the methane, and you can use it for fuel. So it's a valuable resource. Okay. One other place, I don't know if you're familiar with the coast, but there's this stuff called pluff mud. I can't get my pen to write. Uh-oh. Weird. It's not writing. Okay. Well, the pluff mud is the mud that when the tide goes out, you go to Folly Beach and the tide goes out, or Hatteras, since we're in North Carolina, and the tide goes out, that black mud, and the way that it smells when the tide is out, it has that kind of sulfury rotten egg smell. That is, you're smelling the methanogens that live in that pluff mud, and they are decomposers. Okay, so that was a really good lecture. We covered a lot of material. I hope you enjoyed it. Next time we'll talk about reproduction and lots of other interesting topics. Okay. Hope you enjoyed the day. I hope you're enjoying life. Take care of yourselves, and I'll see you next time. Adios. Bye-bye.